afternoon, everyone, and welcome to the 2025 New York ACS Project C Research Symposium. I'm Dr. Ping Ferlin, 2024 Chair of the New York ACS, and I'm honored to serve as your moderator as we celebrate the outstanding achievement of our young STEM researchers. A heartfelt thank you to Dr. Krushna Kluri for his dedication in making this event possible, and to Ms. Nadia Mokar and all our mentors for their invaluable support. We also deeply appreciate our judges for generously sharing their time and expertise today. Ms. Sanhua Zhang, Pacifica School, Los Angeles. Drs. Kat Bay and Emma Kluskowski, Schrodinger, New York. And Dr. Lee Hoffman, Professor of Chemistry, Drexel University, Pennsylvania. Special thanks also to New York ACS, Southern California ACS, and an ACS Local Section Activities Committee grant for co-sponsoring and supporting this symposium. Without further ado, let's begin. Our first speaker today is David Rivera, who will kick off with his research on microplastic transport through porous media, an important study for environmental protection. David, the floor is yours.
Thank you, Dr. Ping. Good afternoon, everyone. My name is David Rivera. I'm a student at the Jose Marti STEM Academy. Over the past summer, I had the privilege of working under the mentorship of Dr. Bufadel through the Project Seed program, which I'm proud to present to you guys today, titled Microplastic Transport and Deposition in Porous Media. Before we begin, I'd like to thank the ACS for providing this opportunity for students to present their research. I'd also like to thank Dr. Nadia Makar and Dr. Mina Armani for helping me with this research and for providing students with this opportunity to engage in research. With this, let's begin. For starters, green infrastructure is a nature-based approach to water management and water mitigation. It's going to mimic systems seen in rain gardens and bioswales, and it is a system that we plan to optimize and implement, which may help filter out microplastics from stormwater runoff. Microplastics are plastic particles less than 5 millimeters in size, which stem from the byproducts of tires, roads, and other sources that may pollute our waterways. So when rain passes, plastic particles that are broken down from tires, such as asphalt and bitumen, are washed from urban areas into drainage systems known as blue-gray infrastructure. When these pass into the drainage systems, they connect to reservoirs, rivers, lakes, oceans, and they contaminate our groundwater. This, in turn, endangers our environmental, aquatic, aerial, and terrestrial animal health. The chart on the right is going to show the microplastic distribution against the most commonly reported sizes, ranging from macro to femto, showing that the most commonly reported sizes are going to be over 100 micron. Our main research objective today is to study the effectiveness the installed system of green infrastructure would have in capturing naturally present microplastics using our naturally present porous media, silicon dioxide, or sand in experimentation. This would in turn help us determine optimal maintenance practices to ensure stormwater runoff cannot contaminate groundwater before entering rivers, lakes, or such as that. Our materials are going to consist of polyethylene microspheres, which are going to be our durable and commonly used microplastic proxy. It is a hydrophobic fluorescent particle with an excitation wavelength of 485 nanometers and an emission wavelength of 525 nanometers. Figure 2 on the right is going to show the molecular structure of our polyethylene polymer. An image of the polyethylene under an electron scanning microscope, and the stock solution we created with our polyethylene and Tween 20. Tween 20, or commonly known as polysorbate 20, is our surfactant solution that we use in experimentation to facilitate the dispersion of the microplastics. This was done as in other solutions, the microplastics, due to their hydrophobic capabilities, would just clump up. Figure 3 on the right is going to show the molecular structure of tween 20. Oh, and it's going to show the molecular structure of the tween 20 and our diluted stock solution. Finally, silicon dioxide, more commonly known as sand, is the media chosen in experimentation due to its purity and its common benefit in the real world. Figure 3b is going to show an image of pure silicon dioxide. Our experimental setup is going to consist of our column chromatography setup, which is our glass column, which our peristaltic pump on the right is going to be calibrated with a flow rate of 2.5 milliliters per minute. Where we're going to set up our columns, where deionized water through the peristaltic pump is going to be pumped through the bottom of the column, where silicon dioxide is going to be added in the top in intervals to simulate a system of the nature and prevent the formation of air bubbles or cracking in the column. 2 columns are then going to be created per experiment with our silicon dioxide media already in the 25 centimeter column. Now, to begin chromatography, our stock solutions are going to be pumped through the system in 3 different intervals. First, we're going to have our equilibrium stage, where surfactant solution is only going to be pumped to equilibrate our system. Second, we're going to have our microplastic stage, where we're going to be passing our polyethylene stock solution through the system to ensure that the silicon dioxide can actually capture microplastics and that our outflowing effluence is clear from microplastics. Finally, we're going to pass the surfactant solution one more time to ensure that the column is washed of microplastics and that the polyethylene doesn't move around in the system. In all 3 stages, we are going to collect effluent samples, and we are going to test them using our Hariba Aqualog spectrofluorometer on the bottom right. Additionally, the media inside of the glass columns is going to be collected as well. And after we dry and we weigh and we dilute them, due to the fact that the Hariba Aqualog spectrofluorometer only tests liquid samples, not dry mass, we are going to collect our supernatant and also analyze the samples using our Hariba Aqualog. The Aqualog tests are first going to be done to standard solutions with known concentrations to create our calibration curves. Our calibration curves are going to be created by using an average of the intensities from 400 to 600 nanometers, which are then going to be plotted against the concentrations of the standard solutions to create our linear equation, which we are going to use against our averaged intensities of our collected effluents and sample intervals to create our concentrations and create our calibration curves. The reason we are going to be using the 400 to 600 nanometers is due to the excitation and emission wavelengths I mentioned earlier of 485 nanometers and 525 nanometers to ensure that we have a good standing for them. Now, additionally, how I said earlier, the presence of sample intervals as dry mass, when we are going to dilute them, we are going to have to multiply this concentration by its diluted mass to ensure we can correct accordingly for their concentrations. The experimental design is going to break down our experiment as I did before. So we're going to have 2 columns per experimentation. We conducted 6 experiments total. We conducted experimentation for 38 micron, 106 micron, and 250 micron. The size that I'm mentioning, 250 micron, 106 micron, and 38 micron, is the size of our polyethylene microplastic. But it is important to note that 250 micron, due to it being such a large particle size, and 38 micron being such a small particle size, is not going to be mentioned today. And 106 micron will be the only data mentioned today. Due to having preliminary results for both 250 and 38 micron, and due to 100 micron being the most commonly reported sizes, as I mentioned earlier. So our results, along with our superimposed graphs, are going to show the complete capture of the 25 centimeter system at only 6 centimeters, as seen in figure 5. Figure 5 shows the concentration of the microplastics in 1 centimeter intervals, proving that green infrastructure is actually able to capture it at 6 centimeters for 106 micron. The calibration curve on the left is used to get the concentrations. Additionally, we did this in the effluents as well. The calibration curve on the left is going to be used against our intensities to receive our microplastic effluents. And figure 7 is going to show the concentration of the polyethylene in our effluents collected during our microplastic pumping. So our results show that microplastic concentration is going to generally decrease with our increasing depth in our 25 centimeter column as shown in figure 5. Additionally, the collected effluents that we have, as exhibited by figure 7, is going to show the complete removal of microplastics by silicon dioxide, as there is no concentration seen in any timed interval of the effluents. Our negative concentration values in both graphs are going to be attributed to complete removal from the related column. These are going to be our results shown in our contour and waterfall plots. On the top, you can see our contour plot and our waterfall plot for our 0.1 gram per liter microplastic concentration standard solution. On the left, you can see the high intensity in its excitation and emission wavelength for the standard solution. However, in our 6 centimeter sample interval that we tested, you see there's no intensity, and that's also proven by our waterfall plots. In conclusion, our negative concentration values in sample intervals and effluents are going to be a complete indication of the complete removal of polyethylene or microplastics by the silicon dioxide, proving that green infrastructure would actually succeed in protecting our groundwater. This proves that utilizing natural resources like sand, soil, can actually create an effective filtration system. These preliminary findings are going to indicate that green infrastructure is an effective method of capturing microplastics. And the photo on the right is a system that we plan to optimize later down the road with additional future work. The future work is actually going to consist of testing different microplastic sizes against different types of medias of different sizes as well, shown by the image on the right as well. This is going to be my bibliography. And finally, thank you all for your time and attention. I do appreciate any questions, comments, and or concerns.
Thanks, David. That was a great presentation.
Thank you very much.
I was going to ask if there are any cons to using silicon dioxide or any reason why it wouldn't be environmentally friendly.
I mean, we haven't really investigated the use of silicon dioxide, but silicon dioxide does show a lot of pros instead of cons, as it is a naturally present for source of porous media. You see it everywhere. Sand is everywhere. There's beaches, and additionally, not only sand, but there's soil. There's other things that can also filter out microplastics. So we do plan on optimizing a system using silicon dioxide and other types of media that we would also have to test.
Thanks. And then just one more. I was wondering if you were looking for kind of a one size fits all filtering, or you would have something unique for every different size microplastic.
Yeah, so that's why we actually plan to test different microplastic sizes against different types of media so that we can see what media affects what type of sizes. As I mentioned earlier, silicon dioxide is not completely capable of capturing 38 micron. It is able to capture it at certain timed intervals, but when time keeps going by, the system is just going to be flushing out microplastics, and I don't have the complete findings of all that. I don't have all the data for it, so I can't really show it and showcase it. But comparing different media sizes is something we would want to focus on doing later down the road.
Okay, thank you.
Thank you very much. Any other questions?
David, there is a question in chat from
Let me see.
Dr. Mary Virginia Orna.
I'm
She
not.
asked, would the capture media become saturated with microplastics?
With the
And
capture,
is there a refreshment method?
I'm sorry. Could you say that one more time? I wasn't
The
able to.
capture media, like your silicon dioxide, would it become saturated with microplastics in time? And if there
Yes.
is a refreshment method.
Yeah. So what we're planning on doing is we're going to use this data to determine optimal maintenance practices for the system. We want to figure out when the media does become saturated and when microplastics are going to start actually penetrating through the system. So when we figure out these maintenance practices, we can figure out how often we would have to replace the silicon dioxide or whatever other media we have inside the system. But along with future work, we do have to figure out other types of media that we could add to actually prevent saturation in the first place, to prevent us having to replenish the system after each time it does rain. So there is additional future work going on for that as well. I don't have all the data for that at the moment, but the media would get saturated after some time period, and it would have to be replenished. The replacement process, I'm not 100% sure how that would work. Any other questions?
Thank you so much, David, for
Thank you
that
very much.
excellent presentation. Okay.
Thank you. I appreciate it very much.
Our next speaker is Ruby Lee, who explores biodegradable plastics derived from milk as a potential solution to plastic waste. Ruby, over to you.
Thank you. Good morning or good afternoon, depending on where you are. My name is Ruby Lee. I'm a junior at Jose Marti STEM Academy, and I want to thank you guys for spending your time and offering me this opportunity. So, today I will be discussing reducing plastic waste using biodegradable plastics derived from milk. Sorry. According to the United Nations, as of 2021, there were around 8.3 trillion tons of plastic waste in landfills worldwide. These plastics are made from polyethylene terephthalate, also known as PET. These plastics are non-biodegradable as when they break down, they release a greenhouse gas, methane, into the atmosphere. This greenhouse gas is one of the main contributors to global warming, a rapidly rising issue in our world. And despite the government's effort to reduce the amount of plastic waste that we have in landfills, only around 8% of plastic was actually recycled in the United States. My research focuses heavily on casein. So, what is casein? Casein is a protein and a polymer that can be found in almost every dairy product. It makes up about,
Ruby, you're not sharing your screen, so we can't actually see the next couple of slides. Just a heads up.
oh, I'm sorry.
No worries.
Um, okay. Can you see it or no?
You need to show the slideshow. You see there's a button on top to right, upper right corner, slideshow.
Why don't you try unsharing and then sharing again?
Okay, let's try that. Can you guys see
Perfect.
it? All right. So do you want me to restart or just continue from where I was?
You may just continue, Ruby.
Okay. So as I said before, my research focuses heavily on casein, a protein and a polymer that can be found in almost every dairy product. It makes up about 80% of the proteins in these dairy products, and such as milk, yogurt, cheese, and much more. This protein has a high heterogeneity, which means that it has a hard time forming alpha helices and beta sheets. However, this is essential for my research, as it can make the casein flexible and moldable. Casein also has acidic properties. For example, if milk were to come in contact with an acid such as vinegar or citrus, the casein proteins will start to form in clumps. And if these protein clumps comes in contact with the basic solutions such as water, the casein clumps will dissolve and return to its original state. So why casein plastic? Many may think that biodegradable plastics do reduce plastic waste and also help the environment. However, that's not all true. While biodegradable plastics do reduce plastic waste, When they break down, they too release methane, the greenhouse gas, into the environment, which is one of the main contributors to global warming. However, casein plastics do not release any carbon into the air. They break down in compost, soil, or even dirt, and the microorganisms in that environment will feed on the carbon assimilation, preventing any carbon to be released into the atmosphere. So for my research, I use petri dishes, hot plates, low-fat 1% milk, whole milk, ethyl alcohol, cheesecloth, and vinegar. When extracting the casein proteins, I heated up the milk, and when it came to a boil, I added vinegar. After stirring, the clumps of casein will start to form, and using a cheesecloth, I separated the casein from the whey, or what is left over from the milk solution. After making sure that both the liquid and the solids were separated, I molded the casein proteins into a petri dish. After molding, I soaked it in ethyl alcohol for 20 minutes to help strengthen the plastic, and then I let it air dry for around 3 to 5 days, depending on its thickness and its size. After using 500 milliliters of each type of milk and 200 milliliters of vinegar in each trial, the fat-free 1% milk was able to produce enough casein for one petri dish mold. As you can tell in the picture, on the left, the fat-free 1% milk is visibly textured, and this meant that it was also harder to mold. However, um, the whole milk casein was able to make two petri dish molds, and as you can tell in the picture, it had a smoother surface, meaning that it was easier to mold. I would also like to note that in the picture, the casein plastics are in various different stages of drying. For example, the fat-free 1% milk had only been drying for about 1 to 2 days, and the whole milk casein plastic had been drying from around 2 to 3 days, and it was almost completely dry. Now, if you recall, the casein plastics were different colors. This was because when the casein dried, the colors had also changed. When the casein was not dried yet, the colors were around a pale white or an off-white. However, when it dried, the colors became more of a pale yellow. The casein plastic also had shrunken as it dried. For example, the whole milk casein plastic went from 8.5 centimeters to 6.8 centimeters in diameter. After the casein plastic dried, I decided to test its durability. Doing various tests, I was able to find that the casein plastics were able to hold around 550 grams of mass and survive a 6 feet drop test without breaking or cracking, despite it only being 6.8 centimeters in diameter and 0.68 centimeters in thickness. So, if a casein plastic of that small size could withstand so much mass and a 6 feet drop test, I wanted to see how much it could hold if it was scaled up to a regular size plate. Using proportions, I calculated that a regular serving plate, about 33 centimeters in diameter, would be able to hold around 2,669 grams in mass, and a typical dinner plate, which is around 27.5 centimeters in diameter, would be able to hold around 2,159 grams in mass. After doing that, I wanted to test the biodegradability of these plastics. In order to do this, I buried it in a mixture of dirt and soil and checked on it over the span of 3 weeks. As you can see in the picture, there are 3 pots. The first pot on the left contains the fat-free 1% casein plastic. The one in the middle had the whole milk casein plastic, and then the one on the right, again, was the whole milk casein plastic just broken down into smaller pieces. During the first week, the temperatures ranged from around 80 to 90 degrees Fahrenheit, and it was also very humid as we were expecting rain. During that week, the casein had become soft and damp, however, still solid. During the second week, it had rained continuously, and the temperatures ranged from around 70 to 90 degrees Fahrenheit. During that week, the casein was no longer solid. It had textures similar to goo or slime, and the rain may have also contributed to speeding up the process for it to break down. During the third week, the temperatures ranged from 70 to 90 degrees Fahrenheit, and it was humid due to the rain from the previous week. The casein had almost completely broken down. There were only about pea-sized amounts of casein left in the pots, and some of it was already gone. However, there are certain limitations to my research. For example, when trying to make a bigger mold, the issue of uneven drying was causing me to be unable to make bigger molds. For example, while the top of the molds were drying, the bottom were not drying. As you can see in figure 2, cracks would start to appear due to this. Another issue was that when the case in plastic comes into contact with water, it will start to dissolve. Overall, case in plastic is a better alternative compared to regular plastics and some biodegradable plastics. It reduces the amount of plastic pollution, thus helping the environment. It doesn't release any carbon into the air, which in turn helps with solving the issue of global warming. Casein is also really easily accessible as it can be found in almost every dairy product, and casein protein is easily extracted with only one ingredient, an acid. Overall, the casein plastic is also tested to be very durable and strong, as well as environmentally friendly and biodegradable. However, my research is far from finished. I want to continue to research the different types of acid when making these casein plastics, as, for example, this time when using vinegar, the casein plastics didn't smell very pleasant. I would also like to look at the chemical structures of the casein plastic, looking at the bonds and the formulas to find out how to make the plastic stronger and more durable. I would also like to explore encasing the casein plastics with biodegradable resin, also known as thermoplastic starch or TPS. This would help make the plastic much more sturdier and waterproof, solving one of the biggest issues when it comes to casein plastics. It would also make it multi-use as people would be able to wash it and reuse it a couple of times before throwing it out or recycling it. And finally, I would also like to look at the cost factors for producing casein plastic when compared to traditional plastics or other biodegradable plastics. Here are my references, and thank you guys for listening.
Okay, we have 3 minutes for Q&A. For
Hi, Ruby. Great job. I have a quick question. What do you think the role of temperature was in the whole chemical process? Because I noticed you use hot milk. Would you get the same result if you used cold milk?
No, I believe it wouldn't be the same since the hot milk would move the molecules faster and allowing for the casein proteins to better bond and better form the casein protein clumps.
Great, thank you.
You're welcome. I have a question from MVO. What would be the feedstock for casein, and is it sustainable? Would the use of casein cut down the availability for food such as milk or other purposes? Um, I believe that casein is very sustainable as it can be found in almost every dairy product, and it would also be it is also high in availability, I'm sorry, because milk is often one of the biggest products that people throw out without finishing. I also believe that farming more cows or farming more animals who do produce these dairy products would also be very helpful. I have a question from Emma. What causes cracking in the casein? Do you think filtering before molding could be helpful? When the casein protein form into clumps, it becomes kind of solid. So I don't think filtering would be possible. The cracking was due to the uneven drying due to the mold. For example, the bottom didn't have any air ventilation, so it was unable to dry, while the top was left open and it was able to dry. And because the casein plastic would shrink as it dried, the top would start to shrink while the bottom was still in its original state. So that's what causes the cracking.
Thanks, Ruby.
You're welcome.
Thank you so much, Ruby. Now our next speaker is Axel Perez, who will present his work on developing a new organoboron building block with promising applications in biologically important compounds. Axel, the stage is yours.
Thank you, Dr. Pink. Good afternoon, everyone. My name is Axel Perez. I'm a junior at the Jose Marti STEM Academy, and today I will be presenting to you my research project regarding the development of a novel organoboron building block for the preparation of biologically important compounds. This research was conducted over the summer at Stevens Institute of Technology, and it was under the guidance of Dr. Abhishek Sharma. Before I begin presenting my project and all the reactions that took place to synthesize the final building block, I would like to introduce you to some terms I will be referring to in my presentation. So these are the molecules that are present in the reactions I will discuss, and these are the abbreviations that I will be referring to when I explain them. So to begin, the fields of medicinal and synthetic organic chemistry are highly dependent on complex molecules. And so as these fields advance, the complex molecules that are used in these fields also must advance. And behind these molecules are smaller components that these molecules are dependent on, and these are called building blocks. So the building blocks actually add the versatility and the usefulness to these molecules. And that's why a gap in the research and something that chemists are always looking to do is find more of these building blocks that can be used to build these molecules, and they can be used in these fields. Now, the rise of boron chemistry can be traced back to the FDA approval of bortezomib, which was a drug that was used to treat a certain type of blood cancer. And in figure 1, you can take a look at the chemical structure of bortezomib, and you can see that it has boron in its chemical structure. So after the FDA approved this drug, chemists took interest in using boron in synthesizing different molecules and different building blocks that could be used in medicinal chemistry. So the versatility of boron comes from its empty p orbital in the sp2 hybridization state, which you can take a look at in figure 2. So this empty p orbital allows boron to bind to form covalent bonds with biologically important molecules such as nucleophiles, nucleic acids, and carbohydrates. And also it provides boron with its high Lewis acidity. Some advantages of boron are also its high specificity, its reduced complexity, and it is also less costly than using other elements or other compounds, with an average cost per gram of $5. Now, the goal of this research project was to synthesize vinyl cyclopropyl diboron as a novel building block, or vinyl cyclopropyl diboron or BCPDB. And the purpose was to produce a new building block to be used as a precursor in organic synthesis. Now, one of the applications for using BCPDB can be observed in figure 3, which shows the regioselective ring opening of BCPDB. In this reaction, BCPDB is reactive with phenolphthalein. In this reaction, BCPDB is reactive with phenolphthalein, producing a homoallylic alpha-alpha diboryl stabilized radical. And this radical can be used as a substrate in the cycloaddition reaction, producing various versions of this radical. And then this radical can be used as an organic precursor in other organic reactions that could be used to access other molecules. For the methodology to access BCPDB, it was a series of three reactions took place that can be observed in figure 4. The first reaction was a dehydrogenation reaction. The second reaction was an alkylation reaction. And the third reaction was an SN2 prime reaction. For the first reaction, BPN-M was dehydrogenated using a base LDA. So a hydrogen atom was removed from one of the carbon atoms in BPN-M. And so this left a negative charge on the BPN-M, which allowed for the second reaction to take place. And this was the alkylation of TdB. And in this reaction followed an SN2 reaction mechanism. And so BPN-M with the negative charge was the nucleophile, and TdB was the electrophile. And also the bromine on the first carbon group of TdB was the leaving group. So BPN-M attacked the TdB and it replaced the bromine group in the TdB. So this allowed them to bond together to produce TAB as the product of this alkylation reaction. And with DAB available, an SN2 prime reaction took place. And so LDA was used again to remove one of the hydrogen atoms from the structure of DAB. And this allowed for the SN2 prime reaction to take place, producing BCPDB as the final product. After the reaction concluded, we obtained the reaction mixture in a series of separation for verification, purification, and identification techniques were performed. So with the reaction mixture, we performed TLCs or thin layer chromatographies to verify that the reaction went on to completion. So all the reactions were consumed and the product was present. After it was confirmed that all reactions were consumed, the reaction mixture was washed and it was worked up using an organic and an aqueous layer. And so the organic layer was obtained and it was powderized for it to be used in flash column chromatography. And the flash column chromatography allowed us to purify the reaction mixture and to obtain a sample that had the most concentration of the compound. After flash column chromatography concluded, we obtained the fractions or the collection tubes that had the highest concentration of the BCPDB, and we used these to perform TLCs again to verify that the product was present. And once we confirmed this, we took the contents of those fractions and we sent them to the proton NMR spectrometer, or the proton nuclear magnetic resonance spectrometer, to identify whether the compound was actually synthesized or not. And after collecting the samples for the proton NMR, we concentrated the contents of these fractions. So for my results, this is the chromatogram that was obtained after the column chromatography. And so the green peak shows the ELS detection. And this is what we mainly focused on. So the highest green peak shows where the concentration of the compound was the highest. And if you take a look at the numbers on the top of the spectrum or the chromatogram, those shows the number of the test tubes or the collection tubes that were labeled for the system, the chromatography system. So we collected test tubes from 27 to 33. And these were the samples that we used for the TLCs after, and then the proton NMR. And this is the spectrum that was obtained from the proton NMR. And after integrating all the protons, we obtained a final number of 32 protons, which resembles the chemical structure BCPDB, which had 30 protons. And the two remaining protons, we determined that it was contamination from the proton NMR samples. Also, although it is difficult to see in the spectrum, there are two symmetrical high peaks, each representing 12 protons, and this represents the two beeping groups in the chemical structure of the BCPDB, each having 12 protons. So with this data, we concluded that BCPDB was synthesized, and throughout the experimentation, we obtained a final yield of 74%. For my conclusion, for For the conclusion, the NMR data suggests the presence of two BP groups in the final structure. And so we confirm that BCPDB was synthesized. BCPDB was synthesized through a series of three reactions. And after the reactions were concluded, we perform a series of purification, identification, separation techniques used for the synthesis and the concentration of BCPDB. Now, BCPDB can be used as an building block for organic synthesis, for example, providing access to stabilized radicals as previously shown. And before the reaction is further performed, it is optimal to ensure proto-optimization to obtain a higher yield of the product. And it would also be interesting to see the alkylation of another halogen butene. So instead of using trans 1,4-dibromo-2-butene, it would be interesting to use, for example, trans 1,4-difluoro-2-butene. Lastly, before mass distributing also of this building block, it's essential to test its toxicity before distributing. Lastly, I would like to thank Mrs. Nadia Makar for this and many other opportunities for students like me who are interested in science and scientific research in specific. And also, I would like to thank Mr. Salas and Dr. Sharma for the guidance throughout this research project, and the postgraduate, graduate, and undergraduate students at the lab over the summer that helped me with the project. These are my references, and I thank you for your attention.
Thank you so much, Axel. So we have 4 minutes for Q&A from our audience.
Thanks, Axel. I could kick it off. I was wondering if you anticipate this reaction would be sensitive to small changes. Like, could you use a variety of reagents and make a full building block library?
Yeah, it's definitely. I think this building block is definitely versatile, so it could be some of the reagents could be changed to obtain the same result or even higher yield for the result.
Did you make any changes when doing this project, or you use the same reagents throughout?
We use the same reagents throughout.
Okay. And then one last question. What kind of percent yield was typical? Do you know?
I think around the same range from 70, from 60 to 70.
Okay. Thank you.
Any other questions? If we don't have any questions, thank you again, Axel. And now we're moving to our next speaker, Lanani Duran, who will discuss how carbon nanotubes and the ferrofluid can help separate microplastics from water. Lanani, please take it away.
All right, hi, sorry guys. All right, so to start off, I would like to thank everyone here today. And my name is Leilani Duran, and I am a junior at Jose Marti STEM Academy. And today I will be presenting to you my project using magnetic carbon nanotubes to remove microplastics from aquatic environments. So to start off, sorry. All right, so to start off, I would like to emphasize and make clear that we are living in the age of plastics. So what I mean by that is that I think that plastic is everywhere. It's woven into our daily lives and we barely even notice. So just take a look around you. The devices that you're holding, the clothes that you're wearing, and even to the packaging of your food that you eat. It's all plastic. This synthetic material has transformed industries, making products cheaper, lighter, and more durable. These products are in our homes, they're in our hospitals, they're even in our cars. But it's all for convenience. Plastic has become an overwhelming force and one we struggle to control. Every year, we produce over 400 million tons of plastic, and only half of it is single use. So it's only used once and then discarded. However, the one thing about plastic is that plastic doesn't disappear. Plastic is a synthetic material made from a wide range of organic polymers such as polyethylene and nylon. So these are considered to be non-biodegradable because the enzymes in the microorganisms that break down these biodegradable materials don't recognize the bonds between the polymers that hold these polymers together. So eventually, even though it may take thousands and thousands of years, they may be able to break down, but in the meanwhile, these plastics will linger in our landfills, they'll clog our oceans, and they'll even break down into microplastics. So, but what are microplastics? I kind of want to emphasize more on that. So microplastics are defined as plastics with a diameter of less than 5 millimeters that are formed through the breakdown of industry. These microplastics are able to go all the way down from nanometer size and can travel through various bodies of water, such as lakes, oceans, and rivers, and can enter said bodies of water through incorrect plastic-based product disposals and other things such as that. So the most commonly found microplastics here could be found are polyethylene and polyethylene terephthalate. So polyethylene is mostly found in bottle caps and detergents, while polyethylene terephthalate, which is also known as PET, is commonly found in plastic bottles, baby wipes, and in some synthetic clothing. So all of these everyday objects contribute to the microplastic pollution throughout many aquatic settings. So one thing that we fail to realize daily is that these microplastics can also have negative health effects to both marine life and humans. There have been many things that have been impacted by us. So we used to use plastic bags every other day. However, we have slowly and surely transformed into paper bags just to make it more environmentally friendly, right? However, we fail to realize how much it also affects us. So these microplastics are able to enter the human body via inhalation, indigestion, and skin contact. And when they enter the human body, they are able to do various damage to us, as they can cause infertility, increase cardiovascular problems, and other things such as that. So me and my mentor wanted to find out how we are able to remove these microplastics if we can't even see them with the human eye, right? So what we did is that we tried to find a method such as nanoabsorbents that were able to take away these microplastics from said aquatic environments. So we attempted to use magnetic carbon nanotubes. But what exactly are magnetic carbon nanotubes? So magnetic carbon nanotubes are synthesized using carbon nanotubes and magnetite, which is also known as bare fluid. Carbon nanotube has a large surface area and are known for the hydrophobic properties, and they're able to bind to the microplastics and can become magnetized using bare fluid. So the combination of two could allow for a highly effective method of massive microplastic removal. However, we would have to test that out. And we decided to go with magnetic carbon nanotube as filtration and other methods seem inadequate for this removal, as filters can only go to a millimeter size and they tend to be more expensive and require more maintenance, requiring more time. While these this nano absorbent require or seem more efficient and more cost effective, and it would be no chemical risk to this. So any chemicals used would not harm the environment at all. So here are my materials that I used throughout this experiment, and my procedure went from, so we used 25 milligrams of bare fluid solution, as well as 25 milligrams of CNT solution. And after that, we would mix them together and sonicate it for at least an hour, and then we would create the magnetic carbon nanotube solution, right? And afterwards, we prepared a plastic water solution which we would mix with the carbon nanotube solution. And afterwards, after sonicating it for an hour, we would have a magnetic carbon nanotube plastic solution. And we would have a really strong magnet on the side of it or the bottom of it. And then afterwards, we would get a clear solution after a couple minutes. And we would decant that. And with that decanted solution, we would heat it up and then see how much plastic there really was. And we also used scanning electron microscopy to really see and visualize how effective it is. Okay. So with the scanning electron microscopy for polyethylene, prior to the treatment, there was a lot of white clumps that you can see here, and those are the microplastics that are inside the water. However, right after the treatment, you can see that most of the magnetic carbon nanotube had efficiently removed most of that plastic. And the same thing applies to to polyethylene terephthalate. So prior to the treatment, there was a bunch of clumps here, as you can see, and that's mostly because of the IMF that is present between these polymers. And afterwards, using the carbon nanotube solution, we can see here that most of it was effectively removed from said treatment. So this is a graph that we also did for that we quantitatively deciphered, as we also like pre-weighed and weighed the amount of microplastic removed and that was still in the beakers. So this is the separation percentage over here on the y-axis, and on the x-axis, this is the amount of microplastic that we use. So you can see here that this graph shows when 1 gram of the microplastic was treated, approximately 99% was separated, indicating that the microplastic capacity is over 4,000. So the carbon nanotube ferrofluid solution is highly effective in smaller concentrations, removing 40 times its mass in microplastics. However, it also shows a decrease in separation efficiency as the microplastic mass increases beyond 2 grams. However, it's still really efficient. As you can see here, it's still 70% over here. So you could still see how it's still really effective. And so for my conclusion, the results indicate that most plastic, PE, and PET tested were effectively absorbed by the carbon nanotube ferrofluid solution. The absorption rate ranges from 94 to 99% across different beaker rates. So the consistency of high absorbent rates suggests that microplastic solutions are highly effective in capturing the microplastic solutions. So for future work, I would like to make a mechanism that could be applied in real life that we would be able to use in like oceans, lakes, and rivers, and to see how much plastic it could actually remove and how effectively it would. And here are my references. And I would like to thank you guys for your time and attention. I would appreciate any comments, suggestions, and everything. Thank you so much.
Thank you, Lenani. So we have several minutes that we can use for Q&A. One question for you, Lenani. For your ferrofluid nanoparticles, did you synthesize them? Did you make those ferrofluid nanoparticles yourself?
Yeah, we would prepare it. Yeah, so what we did is that I would be by myself in the lab with my mentor who was guiding me throughout this process. So I would be making the the ferrofluid solution as well as all the carbon nanotubes were pre-prepared, so it was already bought and all stuff, and then we combined the two.
Did you notice that your ferrofluid nanoparticles turn to rust in water or turn to lose their magnetic properties? Do you experience something like that?
No. However, I did notice when it was left out for a really long period of time,
Mm-hmm.
they would rust. Yeah, they would rust. However, we would do this experiment pretty quickly, so it wouldn't rust. And however, when it was in the water, it wouldn't rust. It was always in a black color, not like in the brownish color that you would see when it would rust.
Thank you, Lanani.
Thank you. And does anyone else have any other questions?
Well, we're a few minutes early, so we're gonna move forward to our next speaker, presenter, Helen Cross. Helen Cross investigates piezoelectric scaffolds and their potential role in spinal cord repair. Helen, over to you.
Hey, so hello, my name is Helen Claus, and ladies and gentlemen, good afternoon, and thank you so much for this great opportunity and for your dedication to spend this Saturday helping us to better our thinking and our research. So to begin, here's my project. So I will be presenting my project on investigating PVDF electrospun scaffolds for facilitating recovery from spinal cord injuries. So to begin with, I would like to introduce a little bit of information about spinal cord injuries. So spinal cord injuries occur when there is damage to the spinal cord, which can lead to a loss of sensation and motor function. This injury could lead to many other side effects and sometimes even death. The injury also I'm also going to introduce DRGs and CSPGs. I'll get into the definitions of those words in a minute. So dorsal root ganglia, also known as DRGs, are sensory neurons that rely on sensory information from the peripheral nervous system to the central nervous system. The injury site also experiences inflammation, and molecules known as chondroitin sulfide proteoglycans, also known as CSPGs, are released. CSPGs attach to the neuron receptors, blocking axon regrowth and stopping the healing process of the spinal cord injuries. Current treatment for spinal cord injuries mainly manage symptoms like pain and aim to prevent further implications, but they provide little to no help with regrowing or restoring function of damaged cells. However, new technologies in biomedical engineering, such as nanofiber scaffolds, are transforming spinal cord injury treatment by encouraging nerve regeneration. These scaffolds mimic the body's natural tissue structure and help cells attach, grow, and repair. And this is exactly what I studied at Columbia University. I used the process of electrospinning to form nanofiber scaffolds. The polymer I used for the solution was electrospun was polyvinyl adenine fluoride trifluoroethylene, also known as PVDF TRFE. For short, I'll be calling it PVDF. And I use this polymer because of its mechanical, biodegradable, and piezoelectric properties. These are all properties that help support neural cell growth. And in this experimentation, dorsal root ganglia, also known as DRGs, were cultured on electrospun PVDF scaffolds and on tissue culture plates in order to compare the effect that PVDF has on cell growth. Additionally, chondroitin sulfide C, also known as CSC, which is a type of CSPG, was added into two groups to determine if its presence could mimic spinal cord injury conditions. And this led to my question. Are PVDF electrospun fibers a suitable scaffold for nerve repair using DRGs under spinal cord injury conditions? And is it possible to demonstrate spinal cord injury conditions using chondroitin sulfide, using CSC to mimic CSPGs released during the spinal cord injury process? And my hypothesis is that the PVDF scaffolds will show a greater DRG neural extension compared to standard tissue culture, and the addition of CSC will mimic the expression of CSPGs during the injury phase. And for my methodology, sorry, I messed up the slide. Sorry. So for my methodology, here are the first steps that I took. So the first process was to make the PVDF solution. So I'm sorry, I got a little confused. So for my first step was to make the PVDF solution. I first combined the PVDF polymer with methyl ethyl ketone, also known as MEK, and I poured this into a glass vial and combined them together. And I placed the vial on a magnetic stir with the heat and stir rate on for about 2 hours. And after that, I placed the sample under a fume hood, so for 8 hours, so for overnight. And then the next process was to electrospin this polymer. So I first turned on my parameters, so my dehumidifier, I set my voltage and flow rate, and I placed a collector into my electrospinning box and connected it to a ground, so my voltage source. Then I poured the solution into a syringe and placed it into my box. And then I connected my voltage source to the needle. So now I have a connection between my collector plate and my needle. And then I turned on my other parameters, such as my exhaust, and then I turned on the flow and voltage rate. And then once the solution was electrospun into fibers, I placed the scaffold on aluminum foil, labeled it, and then I let the sample aerate for 4 days. So here on the bottom, in the third picture, you can see my setup. If you see the collector plate, it's aluminum, and you see those white fibers on them, that's my scaffold forming. And after that, the next process was annealing and quenching my scaffold. So after the solvent had evaporated from the sample, so after my sample had been aerated for 4 days, I placed my sample into the oven. So this is a process of annealing for 96 hours. And then I took the sample out of the oven and placed it into an ice bath. So this is the process of quenching. And then I stored my sample in a piece of aluminum foil. And after, when I was ready to measure my fiber diameters, I took my sample out the foil. I cut a piece of my scaffold and looked at it under a Nikon fluorescence microscope. And I did this in order to help me measure fiber diameter. And so you can see my setup, my microscope setup right here on the two bottom photos. And I use the bright field mode on a Nikon fluorescence microscope to help me see each individual fiber. And I can then I use a computer attached to the microscope to help me, and I use a program called ImageJ to help me measure each fiber diameter. So all I had to do was zoom in into my fibers and then drag from one end of my fiber to the other to measure the fiber diameter. And it is important to measure fiber diameter because it is important for the scaffold to have consistent fiber diameter. It is a scaffold with thick fibers on one end and thinner fibers on one end is just not suitable. It has to be consistent. So I measured my fiber diameter to make sure that my scaffold had consistent fibers. And then after that, I used the Nikon microscope again, and I used it to look at my dorsal root ganglia, so my DRGs, and measure their neural extension to see if they actually grew. So I used the Nikon fluorescence microscope, and then I took images of the DRG bodies and their neuroids, and then I calculated the extension of the neuroids using, again, the program called ImageJ. And then I also use a confocal microscope to get more images and to have better measurements. And then these are the materials that I utilized. And then now onto my data. So the PVDF samples that I tested are listed here on the screen, so right here in the middle. And then to the right, there is a picture of the fibers. And to the right, there's a picture of the fibers under the microscope that I used when I measured the fiber diameter. So here you could see all these little, like, they look like hairs almost. Those are my fibers. And then, so this was the image that I used when I measured my fiber diameters. And then right here to the left, here is a picture of a bar graph. And these are the results that I received after running the ANOVA test. This graph shows the average fiber diameter of the various PVDF samples, and then I'll name the samples right now. So I had samples A, B, and C. Those are the samples that were mine. And then I had my control groups D and E. So the control groups were other scaffolds created by other people in the lab. And the reason that I used those as my control group because I wanted to see what their fiber diameters were compared to mine. And there's no statistically difference between groups A, B, and C. So my groups. So that ensured that my fiber diameters, so that my groups, that my fiber diameters were consistent. So there was no difference in my measure. I mean, there was no different. There is no big significance between my fiber diameters. They were all pretty much consistent. But there is a significant difference between my groups and the control groups, and this could be due to the way that the other people measured their fiber diameters. Their fiber diameters are a bit larger than mine, but this could be due to the fact that their measuring techniques were different than mine. When it comes to measuring fiber diameter, you use a computer and it really goes down to the pixel. So probably when they were measuring their fiber diameters, they could have added extra pixels to the measurement, which could conclude why their measurements are bigger than mine. And then here are the neurofilament immunostaining images of the DRG bodies on PVDF scaffolds and the tissue culture plates. And here are the 4 groups. So we have DRGs on the scaffolds with CSC, DRGs on the scaffolds without CSC. We have DRGs on tissue culture plates with CSC and DRGs on TCP on tissue culture plates with no CSC. And these are the 2 graphs that I used to compare average neural extensions of the DRGs. So graph A shows a statistical significance in neural extension of the culture method, so PVDF versus TCP. And graph B shows statistical significance in neural extension of treatment presence, so CSC and no addition of CSC. And then I'll get into what all this means in a little bit. So for my conclusion, looking back at the immunostain images, I'll pull those up again. Looking back at my immunostaining images, there was more axonal density on the DRGs that were cultured on PVDF scaffolds in comparison to the ones cultured on tissue culture plates. However, the DRGs cultured on PVDF with no addition of CSC had the most axonal density than any other group, which is odd because I expected more axonal growth on the groups with CSC, since CSCs are supposed to mimic spinal cord injury conditions. CSC prevents axonal regrowth, and the purpose of the scaffold is to regenerate growth, but this wasn't seen in the samples. And again, looking at the images, the DRGs cultured on TCP had no growth, which is odd because this probably means one of two things. We didn't take a clear I didn't take a clear picture, or my cell died. There's no axonal growth at all. So when I was probably feeding the cells, I fed the media. When I fed them, I probably punctured the cell and it died, which is why it has no growth at all. And I only had 4 cells to work with, so I didn't have a large sample size. So this really hindered my results. And additionally, the TCP samples with CSC had less neural extension growth than any other group without CSC, which again is odd because I was expecting growth on the group. I wasn't expecting any growth on the groups without CSC because the scaffold wasn't present. I wasn't expecting any growth on the samples that were cultured on TCP because the scaffold wasn't present. And the DRGs were simply on tissue culture plates. There was no scaffold, so I wasn't expecting any growth at all, but there was. And these conflicting results really make it difficult for me to draw any conclusions about the effects of using a scaffold on DRG growth. And in addition, it makes it difficult to assess if CSC is an accurate mimic of CSPGs present in spinal cord injury conditions. And these inconsistent results in small sample size limited the conclusions I could make. And for future experiments, I plan to use larger sample size because that is needed to verify my hypothesis. And lastly, in the future, I plan to investigate the possibility of adding intracellular peptides to the PVDF scaffolds, as they could potentially speed up the spinal cord injury recovery because these peptides are able to regulate pathways in spinal cord injury recovery. And of course, I plan to use a larger sample size again, and I plan to actually redo the research all over again and see if I could obtain better results. And here are my references, and thank you so much for your time.
Thank you so much, Helen. So we could take a few questions from our audience.
Thanks for your presentation, Helen. I think you almost lost me at the procedure. It just seemed very impressive and complex. How much of it did you find in the literature, and how much did you guys kind of develop on your own?
In literature, we found a lot because this actually people have actually tried to do this beforehand. So a lot of this we found in a lot of research papers, especially the electrospinning process. Electrospinning is something that isn't overall that new. Let me pull that up right now. It's not a new process, so it's been done a lot. The only thing that we had to figure out the only thing that I had to figure out myself was which polymer to use, and that was actually kind of easy to do because it was because so many polymers had been investigated. So I knew that I wanted to use PVDF because of its properties and because of its piezoelectric properties. So a lot of it was mostly found in literature.
Thanks. Did anything not work that you had to kind of pivot away from?
Us. For the most part, everything worked. Like my scaffold, it was nearly perfect. Like I said, my fibers were very consistent. I think everything worked for the most part. The only thing that really affected my results were the small sample size. Like I really only had 4 cells to work with, so it was very, very small.
High stakes. Well, thank you. Great presentation.
Yeah, of course.
Is there any reason why you use the MEK methylated ketone? Can any other solvent be used?
Yeah, so MEK is actually it was used to help kind of stretch out my fibers. If you see in this box, my needle, it was it was almost 2 feet apart from my collector. So that MEK helps with the elasticity of my polymer and of my solution. So the reason I used it was to help my fibers stretch out and reach the collector plate. So like if you see like the needle's right here and then the collector is right here. So I had to find something that would help my fibers reach um, the collector plate. And then I use MEK because of its elastic properties.
Any other questions? Thank you again, Helen. Then.
Yeah, thank you. Thank you for spending your time and then listening to me. I'm very grateful for this opportunity. Thank you so much.
Excellent. Okay, we're moving to our next presenter, Dalla Murillo. Dalla will present on axonal injury and astroglial responses in a mouse model of repeated mild traumatic brain injury. Dalla, over to you.
Hi, thank you. I'll share my screen right now. So good afternoon, everyone. My name is Darla Murillo, and I'm from Jose Marti STEM Academy. So before I begin this presentation, I would like to express my gratitude for this amazing opportunity and extend my thanks to all my mentors that helped me along this project. So the title of my project is Axonal Injury and Astroglial Responses in a Mouse Model of Repeated and Mild Traumatic Brain Injury. So to begin, a mild traumatic brain injury happens when there's a hit or jolt to the head, which temporarily disrupts brain functions. Symptoms of a mild TBI can show up immediately or may take hours or even days to appear. Doctors check for physical, cognitive, and sensory symptoms to diagnose this injury, and it is one of the most common types of brain injury, making up 70 to 90% of all TBI cases, and around 42 million people worldwide experience this. After a TBI, a neuron called astrocytes becomes reactive, undergoing a process called astrogliosis, which forms a scar and helps with tissue repair. As I previously mentioned, astrocytes are star-shaped cells that can be found in the brain or spinal cord. They help regulate brain chemicals, maintain ion balance, support and protect neurons, and overall help brain tissue after an injury. To know if astrogliosis is in effect, astrocytes typically increase in number and grow larger. They produce proteins like GFAP, which stands for gliofibrillary acidic proteins, which indicate their activation. What many people don't know is that constant astrocyte activation could cause ongoing inflammation and worsen neurological diseases like Alzheimer's or multiple sclerosis. Which leads me to my hypothesis. Traumatic brain injuries can lead to an increase in astrocytes, resulting in neuroinflammation. This neuroinflammation can contribute to and worsen various neurological diseases. Here are the materials I used. From the materials, I would like to point out the most important ones here, which were the 5-week-old Swiss Webster mice that we used, the stereotactic device, EAE injections, the many antibodies we use like Go Anti-Rat and Go Anti-Rabbit, and of course, the anti-retrieval solutions. Moving forward, here is my methodology. So the first step was to conduct the adult closed head surgery, which is where the mice had received their impact. At 10 weeks old, these mice received their EAE injections. Afterwards, they were later assessed for motor, sensory defects, and their anxiety and depression levels. The mice were then euthanized and their brains were extracted for immunostaining. The immunostaining conducted was specifically for ADK/GFAP, and then after this, imaging and analysis followed. If we look on the right here, this is one of the few pictures that I have of staining, and 2 of them are the behavioral tests that I conducted on the mice. Here is the overall experimental design. So the mice were hit on every 2 days. So they received their first hit at 35 days old, then later at 37, and their last and final hit was given on the 39th day. At 41 days old, we conducted the behavioral test, and then they were later perfused, where, like I mentioned, immunostaining followed. Here is the design of the adult closed head surgery. So as I previously mentioned before in my materials, this is the stereotactic device that was used to give the mice the hit. So they were placed under anesthesia and we deflected their scalp. Then a mechanically driven impactor, 3 millimeters round, impacted the skull at the midline, just rostral to bregma at 3.5 meters per second at a depth of 1 millimeter. There was no gross brain damage, but there was damage to the corpus callosum. Moving on, here is my immunostaining results. So this is the results in gray matter. So in 80K, and so before I begin, sham served as our control group, and TBI was obviously the group of mice that were hit. So in 80K, presented here in the green, there is more 80K production in the sham than TBI. If we look at the GFAP only, there is no significant difference between the sham and TBI. And the 80K GFAP here, we see that there is more production in the sham than TBI. Moving on, here we could say the same results were seen. In the sham group, there was more 80K production than in TBI. There was no significant difference again in GFAP. And then again in the 80K and GFAP results, the sham had presented more. So as like I mentioned, the TBI group showed lower levels of 80K compared to our control group. This reduction may be due to several factors, one of them being that after TBI, it leads to high oxidative stress and inflammation, which could damage cellular components, including 80K, potentially reducing its activities or overall levels. Oftentimes, inflammatory responses associated with the TBI can also release cytokinins and other molecules that interfere with enzyme production. Additionally, TBI also induces cell death and neurodegeneration, resulting in a loss that produces 80K. Consequently, as neurons and other brain cells are damaged, the overall 80K level in the brain decreases. Now moving on to the behavioral data and results. So for the first test conducted, it was called the exit circle test. So each mouse was placed under a clear container and the objective was for the mice to locate the exit. Time for each mouse was taken, and if we look here on the graph on the left, the TBI injured mice took significantly longer than the sham group, suggesting the presence of cognitive defects. Now, continuing on, I would like to extend or explain that in this part of my research, we introduced a second group of mice, which was used to compare the effects of a single large impact to multiple smaller impacts over time. So this second group of mice was hit every 2 weeks. So it was under a 2-week interval compared to the other ones where they were hit in almost a week span every 2 days. So the first test conducted with this group was the horizontal ladder. And this was simply where a mouse walked across a ladder and we analyzed the number of foot slips as they were recorded. The second test involved a device called Rod Road. In this test, the mice were placed on a rotating cylinder that gradually increased its speed, and the duration of each mouse was measured to see how long they could stay on the cylinder before falling, as well as the speed at which they fell off. Here are the results of the exit circle and Rod Road. So in the exit circle test, we see that the sham performed better than the TBI. And this could be due to several reasons. One of them being that obviously they did not experience the cognitive symptoms, but what we see here is by the 6th week, almost the sham and TBI began to converge, showing similar performance. In the Rada Road, despite the TBI experiencing the hit, they continued to perform better than the sham, but there was a slight decline in their time and showing almost again similar performance to the sham. Moving on the right here, we see a video of the horizontal ladder being conducted. So I'll let you guys watch the video, but this is how the mice ran across. So moving on, as we can see here, As the number of footsteps steadily decreased, again, the sham and TBI began to converge in overall results. So this could be due to the mild nature of the injury, which gave the body time to heal, as again, this was done in 2 weeks interval. So the brain ultimately had time to heal before each hit. And this improvement might also show how the body could adapt to smaller repeated stresses, triggering repair and recovery over time. So, in conclusion, repeated mild traumatic brain injury worsens MS by increasing neuroinflammation. Astrocyte reactivity and reduced ADK levels play a key role in accelerating MS pathology. Lower ADK levels following an MTBI disrupt energy metabolism and cellular communication, potentially amplifying this dysfunction. These findings overall highlight the need for therapeutic strategies targeting astrocyte reactivity and ADK regulation in MS patients that are specifically impacted after a TBI. Here's my future discussion. So I would like to continue my project with the activity, with tracking the ADK activity with image day, which will accurately measure particle counts and the ADK intensity. I would also like to do the study with real world parameters. So to model this with more realistic injury forces and intervals between impacts, and obviously a clinical translation where we assess ADK and astrocyte activity in MS patients and link experimental findings to develop therapies targeting ADK and astrocyte regulation. Here are my references. Thank you. Any questions?
We have a chat question. Was the exit circle test or the roll test more difficult to execute, and why?
So the rotor roll test was more difficult to execute due to the fact that the mice, they kept just directly falling. They didn't understand the concept of that they had to keep going on the rotating cylinder. So when we would place the mice on the cylinder, they would just automatically fall to the bottom of the machine. And that would obviously disrupt the results. So we had to almost train them a bit to get used to the rotating cylinder. And as we kept placing each mice in different cylinders, they started to get the hang of it and they were able to last longer. And that's how we were able to get our results. Any other questions?
Have you considered looking at different time intervals for each of these tests? And do you think that would improve the data collection?
So originally we did do the 2 days and then 2 weeks, but we were also thinking of doing maybe intervals with months. But with that would also we would also have to consider that the brain did heal after 2 weeks. So with the intervals of a few months or maybe a few weeks more, we would probably not see that much data as the brain probably did heal by that point. And when we assess like the mice, for example, for behavioral tests, we wouldn't see much of a significant difference, but there's always ways that we could test it out.
Great, thank you.
Any other questions?
Okay, thank you, Dala, for an excellent presentation. And our next presenter is Jasly Arias. Jasly explores DNA nanotechnology for more effective drug delivery systems. Jasly, over to you.
Thank you. I'm going to share my screen right now. Okay. Hi, everyone. My name is Jasly Arias. I'm a current senior at Jose Marti STEM Academy, and I'd like to first thank the American Chemical Society for this amazing opportunity, and I'd also like to thank all my mentors for all the help along this way with this research project. So today I'll be presenting my project on structural DNA nanotechnology, where I will be using nucleic acids as an ideal building block for systemic drug delivery nanostructures. So I think the main question that a lot of people ask me is obviously what is DNA nanotechnology? But if I explain it right away, many people are still a bit confused with this definition, so I'm going to explain it a little bit differently. But instead, I will start by asking, what is the structure of DNA?
And I feel like everyone knows this. It is obviously the double helix structure, and it is formed by Watson-Crick base pairing. And this is a structure that has defined DNA for many years, ever since it was discovered. And so, with this in mind, now we can answer the real question, which is what is DNA nanotechnology? DNA nanotechnology is where we are essentially using the double helix structure to create new structures, which can be seen over here. And DNA technology is essentially where we are recognizing DNA as a primary physical material source for the construction of different nucleic nanostructures. And the reason why this new field is becoming more increasingly discovered and more popular now is because of its many different uses. And the one use I specifically researched was for drug delivery. And the structure that I researched was the tetrahedral DNA nanostructure. And the tetrahedral DNA nanostructure is a pyramidal-based shape. It consists of 4 single-stranded DNA that is formed through self-assembling of its Watson-Crick base pairing. And the reason why this structure is so interesting is because of the fact that it can be used for drug delivery, and it poses many different advantages that other structures do not have. Because in the field of DNA nanotechnology, there are a ton of different structures that exist. So navigating what structure is the best one to use for drug delivery is something that I really wanted to explore. And that's what I did with my project. So for the objective of this project, it was to basically analyze a nucleic nanostructure that has full capability and efficacy of effectively delivering drugs to its target site, as well as evaluating the key traits that the structure has to offer. And so with that in mind, my main hypothesis for this project was that the tetrahedral nucleic nanostructure will showcase a high performance in drug delivery systems and will resolve physical characteristics that enable efficient utility as a drug carrier. So I followed a simple methodology for my research project, which was first to gain atomic force microscopy and gel electrophoresis results. And this will be used to analyze the physical characterization and the structure of the TDN, which is the tetrahedral DNA nanostructure. And then the second portion of the project was to do additional literature review research of studies that have employed the TDN structure to analyze how efficient it was in delivering the drugs. So, for my first piece of evidence, I will be presenting this gel electrophoresis. As you can see here, there are 4 lanes that all include one strand of DNA, and with addition of each lane, there's one more strand added. Now, this is important because this is the first piece of evidence that will define the self-assembly trait of the tetrahedral DNA nanostructure. As you can see, the 4th lane is the one I want to pay the most attention to because it is the one that carries the fully assembled tetrahedral DNA nanostructure. And also, what you need to keep in mind as well, and that we want to pay attention to the most, is the fact that the TDN has approximately 254 base pairs. And this is important because due to its small size, it does provide enough space for small molecules like different types of drugs to be implemented in the structure. And that is something that other nanostructures do not have. With their bigger base pair weight, there's less space to add actual drug molecules into the structures. So that is the first advantage of the TDN structure that is offered. And the second image that I collected was another gel electrophoresis results. This was a page gel, and as you can see here, from lanes 2 to 5, these are all the single-stranded DNA, all the 4 strands. And from lanes 6 to 12, those are all the TDN structures that have been assembled in different concentrations of magnesium. And this is also important because another thing that we want to prove with the tetrahedral DNA nanostructure is the fact that it can withstand any type of environment, because that is also essential for its definition for being used for drug delivery. And as you can see here, all of these results do show that the tetrahedral DNA nanostructure was able to be formed in any and all types of magnesium concentrations. And then my third piece of evidence is the atomic force microscopy imaging. Now, for some context with this, when it comes to DNA nanotechnology and different types of nanostructures, when one person is trying to collect an AFM image, it is very difficult to collect with other types of nanostructures because of how complex the structures are. But because the tetrahedral DNA nanostructure only has 4 single-stranded DNA and its self-assembly is a very simple procedure, I found no difficulty in collecting these results as I would in comparison to other types of nanostructures. So that is also important because this also further proves that the TDN is able to be assembled. All of the little triangular shaped structures that you're seeing here are all tetrahedral DNA nanostructures, and that was captured by the atomic force microscopy. And now for my fourth piece of evidence. As you can see here, these are two qualitative findings for the tetrahedral DNA nanostructure. And the reason why I really wanted to put this into my research was because I wanted to see how the tetrahedral DNA nanostructure interacted and how it delivered the drug to its targeted site. So for these research studies that I found, as you can see here, these are a schematic representation of two drug delivery systems that use doxorubicin, which is a well-known drug that is used for cancer. It's a chemotherapeutic drug. And in this study, it has shown that with the use of the TDN structure, that it was actually able to bypass the efflux rate that would be seen in these cancer cell lines. And this is really important because if you didn't know, doxorubicin, it is an orally administered type of drug. So there are a ton of side effects with this type of administration, which include oral sores, vomiting, nausea, fatigue, and even alopecia. And so by using the TDN nanostructure, we are able to eliminate all those side effects. And that is something that also is why DNA technology is becoming increasingly more used for different types of drug delivery administrations because it is able to eliminate different types of side effects that can be seen with traditional drug delivery systems like pills or any other types. And so with that, all the pieces of evidence that I presented were able to show different traits of the tetrahedral DNA nanostructure, from its self-assembly to its withstanding in any type of environment, and all the way to its efficiency in delivering different types of drugs. I feel like the TDN nanostructure was able to demonstrate that it is a universal nanostructure that can be used for drug delivery, as opposed to other existing nanostructures that are out there as of currently. And I also believe that with the TDN, it is able to be biocompatible and it can minimize these unwanted immune responses or toxic side effects that can be seen with the traditional drug administration systems that many people use today. And is the reason why I confidently feel that people should be looking into it more, especially as a consideration for a new type of drug delivery administration system. And so, in conclusion, the TDN nanostructure can be deemed successful based on its electrophoresis results that have been collected, on its AFM images that have been collected as well, and the two results of the case studies that I have shown. And I also believe that we can anticipate newer studies and more people that are now beginning to look into DNA technology as a new force for drug delivery systems. I think it's important because a lot of people don't know about this field, and I think that's another reason why I really like talking about this project, because it is a new field that many people still do not know, and I do want to bring awareness to it as well. And so in order to expand this research further, my future work would include identifying new areas where I can further improve the tetrahedral DNA nanostructure, because we want to ensure that this structure can be as compatible as it can be if we want to continue to use it for drug delivery administrations. And another thing I would like to do is conduct further studies using the TDN with other types of drug molecules to also further validate the fact that it is a universal compatible drug nanostructure.
And if there's one thing that you want to leave with in this presentation, I think it would be that TDN can be used to change the quality of life for so many people, especially considering the fact that it can eliminate side effects of existing drug administrations, and that change can start today if more people do pay attention to this drug delivery system. So yeah, these are my references for my project, and that will be all. Thank you so much for listening, and I would like to open the floor now to any questions, comments, concerns, or feedback.
Thanks, Jessie. I was going to ask, does the nanostructure's conformation change with different drugs being added for transport?
No, it does not need that. And also, let me go back. For these two studies, with other types of nanostructures, there are different aptamers that can be used to basically kind of indicate the structure to go to a certain targeted site, but that is not needed for this nanostructure. And I feel like that has a lot to do with its tetrahedral shape. So yeah.
And then just one more, can this structure penetrate the blood-brain barrier?
The structure cannot penetrate the blood-brain barrier. Mhm.
Thanks.
You're welcome. Any other questions?
And thank you, Jessie. It must be awesome to actually show the images, atomic force microscope images of these structures. Did you get the images yourself?
Yes, I did
Yes.
it at Rutgers University, in the biomolecular design lab that's offered over there. And I found no difficulty in collecting these results. I first shadowed one of the PhD students that worked there because since it's a new field and I was like my first year doing research, I didn't know how to use these tools, but after seeing them and when I did it, I found no difficulty in doing it. And I think that also speaks volume into how easy this field can be and how it can be implemented for drug delivery nanostructures.
Excellent. Were there particular challenges involved in using the microscope to see something so small at this level?
For me, like I said, I didn't experience any difficulty. The only thing that I was worried about is like I would accidentally break the machine, but I didn't experience that. So it all went well, actually.
Excellent. Thank you, Jessie.
Thank you so much. Any other questions?
What kind of drugs do you visualize this to carry? The synthetic drugs or the monoclonal antibodies or drug DNA molecules or what kind of drugs?
So for the my specific study, I used doxorubicin, but as I saw in other literature reviews, any types of drugs can actually be used with this. So I really would like to further explore that and see like how my results would align with theirs. Yeah. Are there any other questions?
Awesome. If there are no more questions, thank you again, Jessie. And
Thank you so much.
and we're moving to our next presenter, Lizzie Lorzador. Lizzie will talk about her work on optimizing enzyme expression in E. coli for anti-inflammatory research. Lizzie, please share your work. Thank you.
Hello, can you see my screen?
Yes.
Okay. So, hello, my name is Lizzie Lizardo, and I am a senior at Jose Marti STEM Academy. Before we start, I want to apologize for my voice. I do feel a little bit sick. Anyways, I have been a part of Project Seed for about 2 years, and during the summer, I worked with Dr. Yufeng Wei in New Jersey City University. And my project is the comparative optimization of caspase-1 expression in E. coli and enzyme kinetics for anti-inflammation. Inflammation is the body's response to a potential injury or infection, and it often presents itself with pain, swelling, redness, and a loss of function. Inflammation is not a simple process. It is a very complex biological process, and it involves the activation of immune cells and the release of chemical mediators to eliminate harmful bacteria or stimuli to initiate healing in the damaged tissue. However, excessive inflammation can occur and it can lead to long-term deadly diseases. Which leads me to the differences between acute and chronic inflammation. Acute inflammation, also known as normal inflammation, is caused by typical infections or injuries such as cuts, burns, or harmful stimuli such as bacteria. Normal inflammation typically lasts for a couple of hours, a day, or a couple days at most. It removes damaged cells and initiates the healing process, which heals the affected tissue. However, chronic inflammation, known as excessive inflammation, can last for weeks, months, or even years, and it is caused by the long-term exposure to chemical irritants such as radiation, constant infections, or autoimmune diseases, and this can lead to long-term conditions such as diabetic nephropathy and atherosclerosis, which permanently damages the tissue. Around 40% of people with diabetes develop diabetic nephropathy, and hundreds of thousands of deaths are caused by it. The number one cause of death is cardiovascular diseases, which includes heart attacks and strokes. In 2019, around 18 million people died to cardiovascular disease. Atherosclerosis is responsible for nearly 85% of cardiovascular related deaths, and it is the main cause of heart attacks and strokes, therefore accounting for a large majority of deaths. Caspase-1 is a protein essential for the maturation of pro-inflammatory cytokines, including IL-beta and IL-18. Caspase-1 is associated with pyroptosis, which, apart from apoptosis, which is a safe form of cell death. Pyroptosis is an inflammatory cell death that features cell swelling and membrane rupture and the release of pro-inflammatory contents, which ultimately leads to excessive inflammation. Therefore, it is hypothesized that the inhibition or the removal of caspase-1 can prevent chronic or excessive inflammation. As mentioned, the issue is that excessive inflammation can cause very deadly diseases such as cardiovascular diseases, which is the number one cause of death globally. And the purpose of this project is to develop drug inhibitors that reduce this excessive inflammation, which leads me to my hypothesis that the inhibition of caspase-1 will hopefully prevent chronic inflammation. Methodology, terrific broth. This research investigates caspase-1 role in inflammation, focusing on optimizing the expression of its catalytic domain from E. coli cells using two different cultivation methods, which would be manual induction and autoinduction. TB, as shown here in this slide, uses autoinduction. This diagram shows the step-by-step process of bacterial culture and the process and the protein expression of E. coli cells. E. coli was used to E. coli was used because it easily grows and it can easily be manipulated by using simple laboratory equipment. The bacteria is grown in the media and harvested by centrifugation. The high-speed centrifugation is then used for the protein extraction of caspase-1. Using the result from the methodology shown before, I was able to purify the proteins and the chromatograms as shown below, and this displays the UV absorbance peaks of the protein with the purification column. Larger peaks suggest higher or more protein concentration of caspase-1, as you can see in both graphs. By using the samples with the highest protein concentration, I was able to express it in a gel format. Fractions 5, 6, and 8, or samples 5, 6, and 8 are the purest and the most concentrated, whereas fractions 2, 3, and 4 show a lot more impurities. Fractions 9, 12, and 13 are pure, but they are much less concentrated. And by these numbers, we are going left to right. So starting from the first column, which would be 1, all the way to the end, which would be 12. Moving on to the methodology for lysogeny broth, or LB. LB is the manual induction form, and it basically uses the same process as the TB methodology, but it obviously uses some different types of samples or chemicals that are not manually inducted, and these were switched out in order to really see the effects of manual induction. Using the result from the methodology shown before, I was able to purify the proteins, and the chromomatograms, as shown below, displays the UV absorbance peaks of the protein within the purification column. Similar to the TB graphs, larger peaks suggest higher or more protein concentration of caspase 1. As you can see in both graphs, LB has a larger slope than TB, as I showed before. Using the samples with the highest protein concentration, I was able to express it in a gel format. I combined fractions 2, 3, and 4, and 5 into one tube as they were highly concentrated. I combined the rest of them into a different sample. PMPA. This experiment measures the absorbance between the reactions of caspase 1 and PMPA to study the enzyme activity. PMPA is basically a substrate used to optimize caspase 1 and how effectively the enzyme cleaves in the absorbance. Varying PMPA concentrations can lead to the determination of the reaction kinetics and the optimal conditions for caspase 1 growth. These results show the enzyme kinetics of caspase 1 at two different concentrations, one of them being 0.89 and the other one being 0.052 at different PMPA levels. Both graphs show the max velocity and the Km values of the enzyme's max velocity and the substrate affinity in different conditions. This is used to optimize the reaction of caspase 1's efficiency in different concentrations. These calculations explain the results by calculating the catalytic efficiency, or the kcat values, of caspase 1 at two different concentrations, one of them being 0.89 and the other one being 0.0526. Based off of molecular mass and enzyme concentrations, the kcat also represents the turnover rate of caspase 1, which shows how effectively the enzyme's max velocity converts the substrate into the actual product. So to optimize the activity of caspase 1, it's actually crucial to see how it performs under a concentrated substrate. So by using a higher concentration, such as concentrated PMPA, the enzyme kinetic behavior, which is the Vmax, and the substrate affinity, which is the Km, can be determined. As shown from these graphs, these are different types of serial solutions of PMPA and how exactly it reacts over time. So as you can see, the navy blue line is the one with the highest slope, and that is also displayed to the graph on the right. Conclusion. The concentrations of the protein collection from LB and TB were successfully significant for growing a small protein, in this case, caspase 1. The concentration of TB was 0.1192, whereas for LB, it was 0.1528. For the enzyme activity of caspase 1, the kcat values from every methodology were very consistent with one another, which means that although the enzyme concentrations did vary, the affinity remained constant. The PMPA substrate was also examined for caspase 1 interaction as well as what we saw beforehand. So overall, LB was the best growth medium because it was more sufficient for growing a small protein such as caspase 1. Although it took more time because it was manually inducted, it is the best for optimizing the growth conditions for caspase 1, which means that one can actually target it and inhibit it with a potential drug, which leads me to the future applications. For future work, the growth of caspase-1 can be optimized even further to fully investigate and understand its role in inflammatory reactions. Other substrates can also be tested, not just PMPA. The kinetics can also be tested to identify potential suited substrates. Here are my references, and thank you for listening to my presentation. I also want to thank ACS and my mentor, Dr. Yufeng Wei, for this amazing opportunity. So I see one question in the chat. It's, have you considered other growth mediums besides LB and TB? In the lab that I was working at, we used LB and TB because typically those are the most common forms of growth mediums. And we decided to use both LB and TB because that way we were able to see the best growth medium for the caspase-1 because not many studies have actually dealt with caspase-1. So it was it was good to see whether or not either a auto induction or a manual induction would affect its growth. Are there any more questions?
Okay, if we don't have any more questions, thank you again, Lizzie, for that outstanding presentation. We're moving on to our next speaker, Marisha Fernando. Marisha will examine the impact of maternal inflammation on fetal neural development and its link to autism spectrum disorder. Marisha, over to you.
Hi, thank you so much. I'll share my screen now. Can you see my screen?
Yes.
Okay. So before I start, I just want to thank everyone here for making the symposium and many of the research opportunities that we have had possible. So my name is Marisha Fernando, and I am a senior at Jose Marti STEM Academy. So to begin, what if I told you that something as simple as a fever in a pregnant individual could cause lifelong neurological symptoms in the child? This is what I worked to better investigate in my project, where I investigated the impact of maternal inflammation on the development of the fetal brain and the occurrence of autism spectrum disorder. So, autism spectrum disorder can be difficult to diagnose in some cases just because it can significantly differ in the way it presents itself from individual to individual. Like in the name, it has a spectrum of indicators that vary based on what symptom is shown and how severe the symptom is. Despite this, some of the most commonly seen symptoms are sensory aversions, repetitive behaviors, restricted interests, and most importantly, difficulties in communicating and understanding the social interactions of others, which is an essential part of navigating everyday life. According to the CDC, it was found to affect 1 in 36 children in 2020, which is a significantly higher statistic than the 1 in 88 children it was found to affect in 2008, and the 1 in 110 children it was found to affect in 2006. Not only this, but in one study conducted, it was found that the combined costs of medical needs, educational support, adult care, and loss of productivity costs up to a total of $7 trillion in the span of 30 years. So, taking into consideration its rising prevalence and the significant financial burden it presents on our society, it calls to attention the need to better understand the underlying mechanisms within this disease. While autism has been linked to various genetic and environmental influences, its cause is still not well understood. One event that has been associated with an increased development of autism spectrum disorder is maternal immune activation. This can be triggered by things like I mentioned before, fever, allergic reaction, and other kinds of injuries. And in this process, the cells in the body release cytokines known cytokines, which are signaling molecules, in order to prompt this inflammatory response to occur, and one of which is interleukin 6. While there are a number of different cytokines that are already present in the body and released in order for the inflammatory response to occur, interleukin 6 is of interest in this project because it is able to cross the placental barrier. And so once it crosses the placental barrier and enters the fetal blood circulation, it can enter the fetal brain, which can then be a hazard to fetal neurodevelopment. And this is for many reasons, because it can alter gene expression in neural progenitors in the subventricular zone. Neural progenitors are specialized cells that can differentiate to form other cells like astrocytes and oligodendrocytes and other cells that are necessary to form a complete neural circuit. Not only this, but IL-6 can cause an inflammatory cascade where other cytokines are also released in the body, which can further be a hazard to fetal neurodevelopment. So through the assessment of fused forebrain human organoid data that was previously collected, it was found that IL-6 most increased the production of a type of interneuron called a calretinin positive interneuron. And calretinin is a calcium binding protein that's found in interneurons present in the cerebral cortex, hippocampus, and basal ganglia, and it plays a role in the regulation of sensory, motor, and cognitive functions, which is why it's predicted that any changes to the amount of these interneurons found in the brain could possibly lead to many of these symptomatic outcomes seen with neurodevelopment disorders like autism spectrum disorder. So in my project, it was hypothesized that increasing IL-6 production during the period where interneurons are produced in the fetal brain can lead to an increase in the production of the calretinin positive interneurons. And this was tested through the use of a mouse model. So pregnant mice known as dams were injected from day 17 to day 19 of pregnancy, and this period was chosen as this is when the differentiation of neural progenitors occurs, and this corresponds to the third trimester in humans. So the experimental group were given the IL-6 and the control group were given phosphate buffered saline. So once the mice pups were born, 35 days post-birth, they were perfused for the immunofluorescent staining and data collection process to begin. And in this process, the brains were sectioned through the use of a cryostat placed on slides, and then the method of indirect immunofluorescent staining was performed, which is where a primary antibody attaches to the brain tissue and a secondary antibody, which contains the immunofluorescence, attaches to the primary antibody. And this just allows for better observations of the cells when imaging is conducted. The focus of the images that I took was the hilus within the hippocampus, as observation of the entire brain section would show that this is one of the main areas where the interneurons would be present. So each of the brain sections were imaged through 3 different color channels. The blue color channel simply shows the cell nuclei and outline of the hilus. The green color channel shows the calretinin positive interneurons, and the red color channel shows the parvalbumin positive interneurons. These were another type of interneuron that were initially going to be evaluated, but it was found that there wasn't much of a significant difference between the experimental and control groups, which is why the calretinin positive cells are the focus of this research project. So from initial observations, it was seen that brain tissue from IL-6 mice had significantly higher counts of calretinin positive interneurons, which are the tiny green cells in the brain tissue, with IL-6 mice having around 14 cells and the control group having half that amount. And from this, it can be concluded that IL-6, which represents the inflammatory response seen in maternal inflammation, could possibly lead to an increase in calretinin positive interneuron production. And this change in the very delicate balance of inhibitory and excitatory signaling present from the interneurons in the fetal brain can cause impairments in many of the processes needed to form a complete neural system, which is why there are many neurological deficits seen with disorders such as ASD, but knowledge of this presents a possible strategy of developing interventions such as possible medications that could reduce the proliferation of IL-6 and calretinin-positive interneurons. And further study can also direct us towards public health strategies that can reduce risk factors known to cause maternal inflammation, especially in the time period that we live in where there is a rise in emerging infectious diseases such as COVID-19. So going back to medicines that reduce the production of IL-6, there are some that exist today, and they're used in cases such as arthritis and some with symptoms that have been associated with COVID-19. However, their effectiveness has not been used in treating cases of maternal inflammation, but could be a good strategy to use. So as my data collection process continues, there are two aspects that I would alter. One is the type of mice breed used. So in the Neston-Cree TD tomato mice model, what happens is when these mice are injected with a substance called tamoxifen, the Cree recombinase genes remove the stop sequence that exists in the gene that can activate something called the TD tomato fluorescence gene, and the Neston-Cree combination makes sure that this is only activated in the types of cells in the brain that have the nestin protein, like neural progenitors. So through this process, not only are we going to be able to have better estimates of the amount of interneurons that exist in the fetal brain, but also observe different patterns of migration and differentiation starting at the neural progenitors themselves. So looking up at an image of the gel electrophoresis that was performed, the crossing of mice that have the Cree positive gene as well as the TD tomato heterozygous gene would produce the type of mice breed that can make this type of experimentation possible. And another aspect I would change is the method of data collection, where what I previously did was once an image of the brain tissue was taken, I would manually count the amount of cells in the brain, but the process of navigating the microscope could cause possible overlap in the amount of cells that I can see, which can lead to me counting a larger amount of cells than there actually is present. So with the stereology technique, there will be boxes placed on each of the brain tissues by the technology itself. And then through this counting process, it will allow me to have more accurate counts of the amount of cells present in the brain, as well as counts of cells present not only in the tissue, but the entire brain itself. So overall, being able to have more accurate data collection methods, as well as different directions that I can take in evaluating the behavior of neuroprogenitors under the influence of maternal inflammation and IL-6, I hope to have a more definite conclusion on the influence that maternal inflammation has in the development of neurodevelopment disorders. And that brings us to the end of my presentation. Thank you so much. Are there any questions?
Thanks for your presentation. I wanted to ask if there was any non-invasive method to test maternal inflammation that's beyond symptoms.
What do you mean by beyond symptoms? Like
I guess
the behavioral
rather
symptoms?
than simply reporting like a headache, is there a way to kind of test the inflammation and see how it changes with medication?
So the inflammation in the mother itself and not looking at the effects on the mice pups?
Yeah.
I don't think in my case that would really be necessary because the interleukin-6 represents the inflammation that's happening, and our focus is seeing how this affects the mice pups instead of seeing the behavior of the dams. But
okay.
Yeah. Okay.
Thank you.
Okay, are there any other questions for Marisha? Thank you, Marisha, once again for that excellent presentation.
Thank you so much.
We're moving on to our next presenter, Valerie Duran. So Valerie will investigate GABA-A receptors in a cell model, contributing to our understanding of cell signaling. Valerie, the stage is yours.
Thank you.
You
So,
got it.
hello everyone. My name is Valerie Duran. I'm a senior at Jose Marti STEM Academy here in Union City, New Jersey. And I conducted this research under the guidance of Dr. Reed Carroll at New Jersey City University. Here, I investigated type A GABA receptors through the induced autophagy of HEK293 cells. And before I begin, I would actually like to give a quick introduction to autophagy. So, autophagy is an essential biological process that occurs in our bodies. Here, our cells are able to discard any damaged or unneeded organelles and proteins in order to maintain homeostasis and promote cell survival. Autophagy normally occurs when cells start facing very stressful situations such as nutrient or oxygen deprivation. And the image here showcases how the autophagic process works. It begins with a phagophore, which is a double membrane structure that envelops around all of these damaged or unneeded organelles and proteins. Once this phagophore completely closes, it becomes an autophagosome, which is a double membrane vesicle, and this vesicle then moves and fuses with a lysosome, allowing lysosomal enzymes to completely disintegrate all of these inessential components of the cell. After this degradation process, the cell is able to recycle any of these unneeded materials and actually use them to maintain fully functioning cell pathways and systems. Autophagy is also incredibly complex because there are several proteins that are associated with autophagy's regulation, one of which is the gamma aminobutyric acid receptor, more commonly known as GABA receptors. So, GABA receptors are the main inhibitory neurotransmitter receptors of the central nervous system. When GABA, which is an inhibitory neurotransmitter, binds to this receptor, the neuronal activity of the target cell decreases, preventing chemical messages being sent from one neuron to the next. GABA receptors can actually be divided into two subtypes, only one is linked to rapid synaptic inhibition, which is the type A GABA receptor. And here on the images to the right, we see that these types of GABA receptors are ligand-gated ion channels that are often made up of pentameric combinations of alpha, beta, and gamma subunits. Here we also see that there's a chlorine ion pore. So chlorine ions enter this pore or this channel into the neuron, allowing for a concentration gradient that actually leads to the prevention of chemical messages being sent from one neuron to the next. Another protein associated with autophagy and GABA receptors are the GABA receptor associated proteins, also known as GABA-RAP. And there is very limited research regarding GABA-RAP's functions. However, we do know that in connection to autophagy, GABA-RAP is involved in the intracellular trafficking of GABA receptors as well as its surface expression. And they can do this by binding to the gamma 2 subunit of the type A GABA receptor. Similarly to LC3, which is another autophagy marker in the same family as GABA-RAP, it is theorized that these ATGA proteins are involved in some aspect of autophagosome formation, so either in the closure of the phagophore, which then turns into the autophagosome, or in the fusion of the autophagosome to the lysosome. There are also several neurological implications related to disruptions to autophagy and GABAergic processes, such as neurodevelopmental, neuropsychiatric, and neurodegenerative disorders, such as Alzheimer's disease. And in autophagy, when there is some sort of disruption to this process, autophagy is no longer able to discard any of these toxic proteins or organelles that we may see in a cell. And this is what happens in Alzheimer's. There's an accumulation of toxic proteins in neurons like amyloid beta and tau tangles, which causes neuronal death and leads to a decline in memory and cognition, which we see in Alzheimer's patients today. Another part of this is that GABAergic processes are also very important in maintaining excitatory and inhibitory processing in the brain. So when it stops working or there is some sort of dysfunction in that process, there can lead to an imbalance in excitatory and inhibitory signaling, which can lead to grave impacts in the hippocampus of the brain, which is associated with memory storation and formation. It is also concerning to understand Alzheimer's disease as a whole or in a more sociological aspect because there are several statistics that show how grave it is or its impact in our world. For instance, Alzheimer's disease is the leading age-related disease in the United States and the fifth leading cause of death in the world. Again, these are very concerning statistics that we have to keep in mind. So this led me to actually go to the goal of this study. The purpose of my study became to understand the role of autophagy in regulating type A GABA receptors in the gamma 2 subunit through human embryonic kidney cells, which are the HEK293 cells, and then evaluate whether or not GABA-RAP actually links GABA receptors to autophagy. My hypothesis was that within the autophagic process, GABA receptors are specifically targeted and engulfed by autophagosomes for degradation, and that GABA RAP is responsible for trafficking the receptors into autophagosomes and continuing autophagy. In order to complete this experiment, there were three parts that I had to go through, which were transfections, treatments, and fixation. Transfections were implemented as a manner of introducing this plasmid DNA into the HEK293 cells, and this DNA was that of the gamma 2 subunit in the type A GABA receptor, and it was tagged with a green fluorescent protein also known as GFP. After incubating the cells for 24 hours, the next process was to actually give each cell a certain treatment. So the first cell was a control group. The second cell was given starvation media in order to induce autophagy. The third cell was given starvation chloroquine, which was in order to induce autophagy, but then block the last part of autophagy, which is the lysosomal degradation. And then chloroquine was given to the last cell in order to completely stop the lysosomal degradation. The last part was the fixation. So after incubating the cells for another 2 hours, the final step was to wash off any excess materials off of these cells in order to stabilize them for microscope picture taking. Afterwards, immunocytochemistry was actually used in order to insert or bind a specific primary antibody, which was either GABA-RAP or LC3, and observe its localization with a Cy3 secondary antibody, which exhibited a red fluorescence. And we'll actually be seeing these results here. So here we see the GFP GABA receptor co-localization with GABA-RAP. On the left, we see the control group, and on the right, we see the starvation chloroquine treatment. So to take these pictures, I actually used three settings, one of which was FIT-C, and that was used to see the GFP GABA receptor co-localization since it exhibited a green fluorescence. The Cy3 was actually used to see the red fluorescence of either the GABA-RAP or the LC3 in order to see the fluorescence of those proteins. And then the last part was actually it was actually so it was FITC Cy3, and then it was DAPI, which was for standard use. And this is actually really important because then we could see the cell outline and then see the clusterings in each individual cell, see how many GABA receptor clusterings we have in that cell, and then GABA RAP, and then actually see the co-localization between them. So in here for the results, we see that although there was some sort of co-localization between GABA receptor and GABA RAP in each group, we saw it in the highest at starvation chloroquine with 39%. In order to confirm the statistical significance of this graph, I wanted to take a one-way ANOVA test. And here we see that the p-value of this test was 0.00001, which is less than the p-standard value, the p-value standard of 0.05. The graph, along with the ANOVA test, suggests that the GABA RAPs regulate GABA receptors during autophagy, and that this link is strengthened when lysosomal degradation is blocked, since we use starvation chloroquine, and we actually saw the highest percent colocalization here. Here in the next results, we see GFP receptor colocalization with LC3. On the left, we see the control group, and on the right, we see starvation chloroquine once again. And here are our results for this colocalization percentage. Again, throughout all the groups, we actually see different treatments having different types of or levels of colocalization. They're all relatively similar as we see that the error bars are overlapping with each other. So then I wanted to confirm whether or not this test was statistically insignificant with this graph. And I was able to do this again through a one-way ANOVA test, which showed that the p-value was 0.26. This is higher than the standard p-value of 0.05, showing that although we do have co-localization between GABA receptors and LC3, there is no strong link identified between GABA receptor co-localization with LC3. The last set of data that I took was actually the average GABA RAP and LC3 clusterings. Here we compare the different groups between average number of clusterings per cell between control starvation, starvation chloroquine, and chloroquine. In the three ANOVA tests that I took, I saw that there were different statistical significant levels between average GABA RAP clusterings, then between statistical significances in average LC3 clusterings, and then the last part I wanted to see was whether or not there was a statistical difference between average GABA RAP and average LC3 clusterings in starvation and starvation chloroquine specifically. So one thing that I observed that was, I think, really interesting here was that when the GABA RAP clusterings were low in starvation, it was much higher for LC3 clusterings in that same treatment, and then the opposite effect occurred in the starvation chloroquine, where GABA RAP was actually higher, but then it was lower in the LC3. I decided to take a p-value for that one through the ANOVA test, and then I found that that p-value was 0.04, which is although similar, it is still lower than the 0.05 standard p-value of this test, showing that there is some sort of difference between the LC3 dependent autophagosomes and then the GABA rap and GABA receptor movement. In conclusion, starvation chloroquine was actually concluded to induce autophagy in HEC-293 cells. The heightened colocalization of GABA receptors with GABA-RAP, especially under starvation chloroquine, demonstrated that type A GABA receptors are in fact regulated during autophagy. The lower colocalization of GABA receptors with LC3 in comparison does suggest that the transportation of GABA-RAP and by association GABA receptors occurs in a process opposite to these LC3 proteins. And again, it also shows this or a different explanation for the starvation and starvation chloroquine differences that we see here. Another thing that I did observe was actually the altered distribution of GABA-RAP throughout all these treatments in the average clusterings, which shows that there is some sort of movement occurring between GABA receptors with autophagy and specifically autophagosomes controlled by LC3. The larger average of GABA-RAP clusterings also suggested to us that, again, it solidified this idea that GABA receptors are trafficked by GABA-RAP into autophagosomes. For future research, I do believe that there is more evidence needed to investigate autophagy's involvement in managing GABA receptor dynamics. Some experimentation variables I do want to change in order to understand GABA-RAP specifically is actually using endogenous cells to view GABA-RAP and LC3 clusterings, and then understand the relationship between the two proteins and see GABA receptor movement specifically within autophagy. So seeing how exactly the GABA receptor is moving throughout this process. And once again, my name is Valerie Durand, and my project revolves around the investigation of type A GABA receptors through the induced autophagy of HEK293 cells. Thank you.
Very good. Any questions for Valerie?
I just saw one in the chat. So, what was most surprising aspect of the clustering results? I actually mentioned this. I think the most interesting aspect, I'll go back to it. I'll actually share my screen again. There we go. Let me see. And here in this result, actually, the most interesting part was the fact that the GABA rap and GABA receptor movement was actually opposite to the LC3 dependent autophagosomes. So it could suggest that there's something else going on, or that GABA rap, like GABA rap's function stops at the autophagosome fusion, and that after that we don't really see what's going on. So that's why I would like to investigate LC3 and GABA rap relationship more specifically. Thank you for that question. Does anyone else have a question?
Okay, thank you again, Valerie. Okay, we're moving on to our next speaker, Jocelyn Passu. Jocelyn will explore how tree species respond to soil moisture and the effects of climate change on canopy transpiration. Jocelyn, over to you.
Thank you. Hi, everyone. My name is Jocelyn. I'm a senior at Jose Marti STEM Academy. I would like to express my gratitude for everyone who is part of the association and allowed me to showcase my project, which is canopy transpiration in different tree species and their response to soil moisture in a changing climate. Now, it can be noticed all around us that global warming and climate change has been continuing to rise, and it's causing droughts and unbelievable amounts of heat waves, and it's affecting trees. And if it's affecting trees, it's affecting all of us humans, animals, and nature, and it's important to find solutions on how we can restore and stabilize our environment. Now, in the background, transpiration is the movement of water from the trees being released into the atmosphere. And canopy transpiration, which is also known as sap flow, is a process that trees rely on for certain activities to take place, such as photosynthesis and regulating temperatures. And they can be driven by many environmental parameters. The one that I'm mostly focused on was soil moisture, which is the amount of water in the soil. It can be seen here in the picture that the trees, they gain they absorb the water from the soil through the roots and through the roots to the stem, and then that water evaporates out through the leaves. Now, my objective was to calculate and analyze the rate of sap flow by measuring sap flow extensity and sapwood area in two specific tree species, which was black walnut and sugar maple. and studied the effect of soil moisture and the amount of transpiration from a specific time span, which was from early June to late September, because that is where sap flow is occurring for years 2021 and 2023. My hypothesis was that if soil moisture continues to increase, so will canopy transpiration rates, affecting how much water is utilized up in a tree. Now, there are different site locations, which means different data, but the two sites that I mostly focused on was Coffinwoods and Hay Barn. Now, in doing sap flow measurements, two similar sections of the tree bark is removed to expose sapwood, and 1 centimeter to 2 centimeter holes are drilled, depending on the tree's width, are drilled to insert thermal probe sensors, which are previously handmade in the lab, and they help detect sap flow. They're covered by a tin foil to protect them from any heavy rainfall or any wildlife. Now, all these sap flow, thermal probe sensors are all connected to the data logger, which are powered by a solar panel. And through the data logger, it helps record and saves all the data, which are then extracted into raw files and then placed into a baseliner application to be analyzed, which shows the temperature difference from the heating probe and the reference probe sensors. And this shows what a preferred baseliner application looks like from the data being recorded from early June to late September. And this just shows the not preferred version, which just shows no data. Now, in part 2, which is sapwood area measurements, using a holograph increment border at breast height, it can help extract core samples, and the measurements for both bark and sapwood depth are recorded because that is where sap flow is occurring. And the diameter of the tree trunk is measured and recorded just using a regular diameter tape. Now, there are around 2 to 4 soil moisture sensors in each site that connect to the data logger. And something crucial to mention is threshold response. On my literature findings, it was found that at higher temperatures, there can be an increase in rates of transpiration, which can induce plant water stress, which just means that the tree's leaves can decay or wither away or just lose their color overall. And something else to also mention is that 2023 was the warmest year recorded by far. Now, sugar maple, it is a diffuse porous species, meaning they have small pores. So they have less efficiency of water transport going on compared to other tree species. They have a low vulnerability in a drought season, so it does prevent them from having a greater water storage capacity. Now, these charts show that in 2021 for sugar maple, for coffinwoods, it does have significantly more data points compared to in hay barn, because in coffinwoods, it seemed to absorb more moisture from the soil. There was more heavy rainfall in coffinwoods compared to hay barn. Since sugar maple is able to limit its use of water, it doesn't have that big of an increase in its rates of sap flow. Here for coffinwoods, it does have a steady rate of trend, and in hay barn, there seems to be a decrease. For black walnut, it is a semi-ring porous species, so it combines both small and large pores. So they do have a better efficiency of water transport going on. They have a low drought tolerance, so they're not able to limit their water utilization as much. For here in 2021 in black walnut, it can be seen that in coffinwoods, again, it's the same idea for sugar maple. It has more data points compared to in hay barn. For black walnut, it does have that increased rate of transpiration in coffinwoods and hay barn, because since it's not able to limit its water use, it's going to have higher rates of sap flow than sugar maple. And in hay barn, it can be seen that for both sugar maple and black walnut, they do have that threshold response where trees collect moisture from the soil for a certain amount. That's when they begin to transpire. Now, looking and comparing years 2021 and 2023, it can be seen that significantly for sugar maple and black walnut, in 2023, there were less data points. Again, because since 2023 was the warmest year, the hot temperatures were drying up the soils. There was not a lot of heavy rainfall, so trees were not able to absorb that big much amount of water as it did comparably in 2021. Looking on the left side for sugar maple, it can be seen that there is less data points. Again, since sugar maple, it does limit its water use and it does have a small storage capacity. It's not going to have that much of sap flow going on compared to black walnut. It does have more data points in 2023 because it is going to use as much water as it wants to for the rate of sap flow to occur. So this just shows that in 2023, it did have lower amounts of data points compared to 2021. Now, to conclude, environmental parameters such as soil moisture does have a huge effect on sap flow stability, and many different characteristics come into play when it comes to looking at sugar maple and black walnut's relationship, such as different seasons or humidity. And because of increased temperatures, it affects the soil's temperature, reducing in soil moisture, causing higher rates of sap flow, which does increase water stress for trees in the near future. In Haybarn, oh sorry, in Haybarn, there seemed to be less water in the soil, affecting trend of sap flow to be significantly different than it did in Kaufman Woods. And as temperatures continue to rise, it's affecting the amount of soil moisture to create unstable trends of transpiration. Now, something that I want to mention is that in 2020, when I was doing my project, 2023 was the warmest year recorded. But now that 2024 is over, it was recorded that 2024 was the warmest year ever recorded. And it just shows that as global warming is continuing, it will continue to disrupt the tree sap flow, affecting them to have increased stress more early on than predicted. Now, challenges were no data being recorded due to sensors being damaged by weather or wildlife. When I was out there on the field sites, there was, as here in the picture, there was these mice that were where the thermal probe sensors were, and they were eating the wires, and it prevented no data being recorded at all. So we would have to be constantly checking or replacing thermal probe sensors to make sure that we had data being recorded. Now, future studies that wish to be conducted is changing or adding more site locations for larger samples to be collected and recorded, or focusing on different types of tree species and how they may have a different relationship with soil moisture. In this project, I did do diffuse pore species and semi-ring pore species, so later on I can see how ring pore species may have that different type of relationship with soil moisture. And it's important to know that drivers of soil moisture and of canopy transpiration can help and determine the effects of carbon recycling in the future, since it was seen analyzing the data helps how the relationship of canopy transpiration and soil moisture may affect future predictions on ecosystem dynamics and climates. And as the act of climate change is continuing to rise, it has created various impacts on the environment and adaptability of various tree species, and difficulties arise for trees and plants to adapt and remain in one providence. So, as these many environmental parameters come into play, it affects how trees have been able to transpire. Therefore, climate change and the change in the amount and rate of how much transpiration that occurs in the trees affect how stable they can survive in certain environments. Which just goes to show that all trees are important and they help our environment, and we have to help our trees from being disrupted by the impacts of climate change. These are my references, and my acknowledgements go out to the Shaper Lab where I participated in and doing my project, and teachers, and thank you so much for your time and attention. I'd be happy to answer any questions.
Thanks for your presentation. I was wondering how the slow sap growth impacts the tree growth overall. I know you mentioned it briefly and kind of their rate of photosynthesis, maybe.
Photosynthesis, you said?
Yeah, I was wondering how, so the sap flow is kind of
Mhm.
your indicator, right? If there's a problem with the tree and if there's the impact of climate change. Do you expect this to significantly slow how quickly the tree can grow?
So that comes into play with other stuff. I didn't really focus on that. I focused more on on their transpiration rate, so I wasn't able to see like how it would affect them to grow as much. But it's definitely something that I can look into the future.
Okay, thank you for your presentation.
Mhm. Any more questions?
I guess I could go ahead and ask one more.
Mhm.
Why measure the sap flow instead of water concentration directly? Is it a little bit easier?
Um, so when I was doing the project where I on the field site in Duke Farms, they wanted to see more of sap flow going on because where they were doing it, there was a lot of carbon dioxide content that was increasing, so it was better to show the relationship of sap flow more in depth.
Okay, thank you.
Mhm.
Okay. Thank you again, Jocelyn. Excellent.
Thank you.
Finally, Jay Castillo, our last speaker. Jay will present her research on antimicrobial composite materials which could revolutionize infection control. Jay, please share your research.
Good afternoon. Thank you for having me. My research is on the development, testing, and performance analysis of an antimicrobial composite material. And I've done this under the mentorship of Dr. Clively. So my initial interest for this experiment came from learning about the significant problem of bacterial contamination within everyday product surfaces, food safety, and even in healthcare, leading to the transmission of various infections and diseases. And within the American Journal of Infection Control, it was found that 20% of healthcare-associated infections were linked to contaminated surfaces. And a study by the University of Arizona found that 75% of public surfaces were contaminated by potentially harmful bacteria. And this study focuses on E. coli and staph bacteria due to their significant presence in both environmental and clinical settings. And zinc parathion is a chemical known for its strong antibacterial properties and is very commonly used in anti-dandruff shampoos and sprays. And studies have shown that zinc parathion can disrupt microbial cell membranes, inhibit enzyme activity, and interfere with cellular metabolism, ultimately leading to cell death. My hypothesis for this experiment was that zinc parathion infused composite samples would be able to show significant antimicrobial activity. So to begin my methodology, I first began by making the composite samples themselves, and I used both epoxy and polyolefin powder, and they were poured into thin layers and used as a control. And then additional batches were made by then infusing small amounts of zinc parathion into each of these base materials. And once everything was cured, a sterile hole punch was used to create small uniform samples. And then I made the bacterial plates, in which I made an agar mixture, and it was prepared and poured into sterile petri dishes and left to solidify. Each plate was then labeled, and small amounts of E. coli and staph bacteria were streaked onto the surface of these agar plates. And finally, I conducted a Kirby-Bauer test, in which one disc from each of these samples were placed onto the surface of the agar plates with a no disc plate made for both the E. coli and staph bacteria. And they were left incubating for 24 hours, and after checking for the inhibition zones, the process was repeated 3 times to confirm my results. Here you can see the preparation of the agar mixture used for the bacteria plating, as well as the staph and E. coli cultures used to streak the plates. So looking at the results of the material samples on the polyolefin powder, I saw an inhibition zone of around 30 milliliters on both the SA and EC bacterial plates, showing quick and crucial antibacterial activity. As you can see, this significantly opposes the control group's results, as you can see with 0 inhibition zones. And here we can see the results of the base epoxy material with the experiment disc, leaving an inhibition zone of around 4 milliliters, while the control group exhibits none. And lastly, on the E. coli bacterial plates, we can see an area of 2 milliliters on the inhibition here on the right, while the control group, again, no antibacterial properties. So in conclusion, zinc parathion greatly reduces bacterial growth in both thermoplastic, the polyolefin powder, and thermal setting, the epoxy materials. It quickly inhibited bacterial growth in both the polyolefin powder with a 30 millimeter inhibition zone and showed long-lasting antibacterial effects in the epoxy, making it a useful additive for improving hygiene in various materials in multiple settings. And looking for my future research, I will dive into industrial manufacturing, in which I would collaborate with an industry partner that would produce test samples using injection molding machine. And again, I will test the antimicrobial properties. And these are my results. And thank you for listening. I will be open to any questions.
Hi, Jade. I was kind of curious on what part of this process you enjoyed the most and what part you are excited about working on in the future.
This was my first time working with bacteria like whatsoever, so I had fun streaking and making the agar plates. I thought that was really fun and makes me more interested in learning more about microbiology itself.
That's great to hear. Thanks.
Oh, I see the question. Were there other tests you're considering? So the ones you presented. Like if I was going to do anything but the Kirby-Bauer test? Oh no, I was just mostly focused on the Kirby-Bauer test and learning how to do that and what was that about, and just focusing on the antibacterial properties of it. Thank you.
Jay, very nice presentation. I have a question. How did you make your composite material, and what's the ratio? How much of your antimicrobial agent, zinc PT, that you use for making your composite, like for creating that kind of zone of inhibition, right? How much of your ZPT is involved?
Yeah, so I did that based off the ratios of both the epoxy and polyolefin powder I did, so it just changed based off how much of each that I use. But relatively, I used about like 1 tablespoon of the zinc pyrithione powder itself. And I just mixed it into when I was making each of the base materials of it.
How did you make this composite? Through heating, through any other process?
No, it was just the mixture of the powders itself. But for the polyolefin powder, it did have to be heated to make the sample solidify and become the plastic itself. Because before it was just the polyolefin powder itself.
Okay, thank you. Thank you, Jay.
Thank you.
Any other questions for Jay? Okay, thank you, Jay, again, and all our speakers for your incredible work today. What an inspiring afternoon. Your dedication to advancing science is truly commendable. A huge thank you to Dr. Kalori once again, and to our mentors, sponsors, and the judges for making this event possible. Stay tuned for the announcement of our top 5 presenters who will receive awards for their outstanding research. Thank you all for joining us today, and have a wonderful day.